A Denisovan survival gene still lives in Tibetans, long after Denisovans vanished. That is the clue that should not have been there. On the Tibetan plateau, where villages sit higher than many airplane cabins are pressurized, thin air pushes the human body into emergency mode. The lungs pull harder, the heart works longer. The bone marrow starts making extra red blood cells, trying to move more oxygen through a world that barely offers enough of it. For many lowlanders, that response can turn dangerous. Blood thickens, pressure rises inside the vessels. The heart has to push heavier fluid through the body, day after day, night after night, while the oxygen meter still looks wrong. But many Tibetans do not follow that pattern. Their bodies often handle high altitude without producing the same heavy overload of hemoglobin. The old medical explanation sounded simple. Oxygen is low, so the body adapts. Then, scientists looked at the gene called EPAS1. The version found at high frequency in Tibetans did not look like ordinary modern human inheritance. It looked older. It looked out of place. And when researchers traced the signal backward, it pointed toward a population most people knew only from a cave bone in Siberia. The first model broke there. This was not a legend about mountain endurance. It was not a story about willpower, toughness, or some mystical bond with altitude. It was a biological clue, sitting inside the oxygen system of living people, carrying a signature from extinct humans related to the Denisovans. The body had solved thin air in a stranger way than anyone expected. EPAS1 is part of the body's oxygen sensing machinery. Scientists sometimes call it a master switch in the response to hypoxia, the condition that happens when tissues are not getting enough oxygen. When air gets thin, the body has to decide how hard to react. One familiar reaction is to make more red blood cells. Those cells carry hemoglobin, the protein that holds oxygen and moves it through the bloodstream. At sea level, the system works so quietly that most people never think about it. It is just a number on a hospital form, printed beside a normal range. At high altitude, that number can become a survival problem. If the body raises hemoglobin, it can carry more oxygen. That sounds useful, but there is a price. Too many red blood cells can make the blood thick and slow. The heart may strain, circulation can suffer, pregnancy can become harder. Long-term altitude sickness can appear in people whose bodies are fighting the mountain with the wrong tool. On the Tibetan plateau, researchers noticed something unusual. Many Tibetans living at high elevation had lower hemoglobin levels than expected, compared with lowland people exposed to the same thin air. Their bodies were not just flooding the blood with red cells, they seemed to be using another strategy. In the early 2000s, genome-wide studies compared Tibetan highland populations with lower altitude groups, including Han Chinese populations. Genome-wide means scientists were not starting with one favorite gene. They scanned many parts of the genome, looking for places where natural selection had left a sharp fingerprint. EPAS1 stood out. The signal was too strong to ignore. In Tibetans, certain versions of EPAS1 appeared far more often than chance would predict. The gene was connected to hemoglobin regulation, and the pattern matched the medical observation from the plateau. The DNA and the blood readings were pointing at the same mystery. The caveat matters here. This is not a measure of superiority. It is not purity, destiny, intelligence, moral worth, or a ranking of human groups. It is one inherited adaptation shaped by a harsh environment. A useful gene in one place can be irrelevant somewhere else, or even harmful under different conditions. Genetics does not hand out human value. It records survival pressures. That distinction matters because the Tibetan EPAS1 story sits close to dangerous misunderstandings. It involves ancestry, adaptation, and comparison between populations. But the responsible question is narrow. How did one high-altitude population inherit a version of an oxygen gene that helped many bodies handle thin air without the same blood-thickening response? Once scientists mapped where that EPAS1 pattern appeared, oxygen alone no longer explained it. Picture the plateau as a high, cold map. Mountain passes cut through stone. Settlements hold to valleys where crops grow under a hard sky. Yak herding routes cross open ground where visitors may feel breathless after only a few steps. In clinics, blood samples can show how much hemoglobin is moving through a person's veins. In labs, the same story appears as letters on a screen. The altitude is obvious. The genetic pattern is not. Tibetans are not the only people who have lived high above sea level. Andean populations in South America have long histories in the mountains. Ethiopian highlanders have their own deep adaptation to elevation. Each group faced thin air, but their bodies did not all solve the problem the same way. That is the first important break in the simple story. If there were one universal high-altitude gene, scientists would expect the same solution to appear again and again, wherever humans settled above the clouds. The genome did not show that. Andean highlanders often show a different pattern, including higher hemoglobin responses in many cases. Ethiopian highlanders show another pattern again, with their own genetic signals and physiological strategies. The mountain was similar, the solutions were not. In Tibetans, the EPAS1 signal was unusually strong. The adaptive haplotype, meaning a stretch of DNA inherited as a block, appeared at high frequency in Tibetan populations. In many neighboring lowland groups, including Han Chinese comparison groups, it was rare or absent at the same level. That difference told scientists that natural selection had probably acted hard and fast on the plateau. A haplotype is easier to picture as a paragraph copied through generations. Single genetic letters can change one by one. A haplotype is a longer passage that tends to travel together from parent to child. If a useful version helps people survive or have more surviving children, that whole paragraph can become more common in a population over time. EPAS1 looked like that kind of paragraph. Natural selection could explain why the variant spread among Tibetans. The plateau is one of the harshest human environments on Earth. If a genetic variant made pregnancy safer, oxygen regulation steadier, or daily survival easier at elevation, then over many generations it could rise quickly. But selection alone could not explain the deeper problem. The DNA block did not look like a small, modern mutation that had appeared recently in Tibetans and then spread. It looked unusually divergent. That means it carried differences that seemed to separate it from ordinary modern human variation by a deeper stretch of time than expected. The evidence points in a narrower direction. On the map, the gene looked Tibetan because Tibetans carried it in large numbers. In the deeper genome, it looked as if it came from somewhere older than Tibetan history, older than the current population boundaries, older than the familiar story of modern humans walking into new lands and adapting by themselves. The map pointed backward, past kingdoms, languages, and written memory. It pointed toward a population no one had seen alive. Denisovans entered science through a fragment small enough to fit in a hand. In Denisova Cave in the Altai Mountains of Siberia, archaeologists recovered a finger bone and later teeth from ancient humans who did not fit neatly into the known categories. The cave itself became a genetic witness box, holding remains from different human groups across deep time. Around 2010, researchers at the Max Planck Institute sequenced DNA from that tiny finger bone. The result changed the human family tree. The genome was not Neanderthal. It was not modern human. It belonged to a separate archaic human population, related to Neanderthals, but distinct from them. They became known as Denisovans. At first, Denisovans were almost invisible. There was no complete skeleton standing in a museum case. No long row of skulls gave the public a familiar face. For most people, the Denisovan world began as genetic data from a cave. A finger bone, a tooth, a sequence of ancient DNA read from material that had survived in cold stone. That was enough. Ancient DNA can turn a fragment into a population. It can compare one genome against another and ask who shared ancestry with whom. When scientists compared the Denisovan genome with living people, they found traces of Denisovan-related ancestry in parts of Asia and Oceania, especially among some Papuan and indigenous Australian populations. The vanished group had not stayed sealed inside the past. The genome kept one more record. In 2014, a study published in Nature connected the Tibetan EPAS1 haplotype to Denisovan-related ancestry. Researchers compared the unusual Tibetan DNA block with genomes from modern populations and with archaic human DNA. The match that stood out was not ordinary modern human variation. It was Denisovan-like. The survival signal in Tibetans closely resembled DNA from the Denisovan branch. That was the partial payoff. The strange EPAS1 variant was not just a local Tibetan oddity. It appeared to be an inherited fragment from archaic humans related to Denisovans, preserved in modern people because it helped the body handle oxygen on the plateau. Think about that in physical terms. A person walks across a high pass in Tibet. Their breathing changes, their blood chemistry responds. A gene helps regulate that response. And part of the useful version of that gene may trace back to contact between modern humans and an extinct human population tens of thousands of years ago. The old oxygen story was too small. It could explain why adaptation mattered. It could not explain why the genetic solution looked like a message from another branch of humanity. It could not explain why a modern, high-altitude adaptation carried the shape of a Denisovan-related inheritance. That seemed to solve the mystery, until it opened a stranger one. Denisovans were not supposed to be living inside a modern highland adaptation. For a long time, the textbook version of human origins was drawn like a clean tree. Modern humans emerged in Africa, Neanderthals occupied parts of Europe and Western Asia. Denisovans, once discovered, became another extinct branch somewhere in Asia. The branches separated, some vanished. One branch, ours, survived. That picture was useful. It was also too clean. As ancient DNA improved, the branches began crossing. Modern humans leaving Africa did not move through an empty world. They met other human groups. Sometimes they competed, sometimes they replaced them, and sometimes they had children with them. This process is called Archaic Introgression. The phrase sounds technical, but the idea is direct. Archaic human DNA entered modern human populations through interbreeding, then traveled forward through descendants. If a fragment was neutral, it might drift quietly. If it was harmful, selection could reduce it. If it was useful, the genome might keep it. Neanderthal ancestry is the familiar example. Many people outside Africa carry small amounts of Neanderthal DNA. Those fragments are not evenly spread across the genome, and they do not make anyone Neanderthal in identity or culture. They are surviving pieces of ancient contact. Denisovan ancestry is less visible because the fossil record is thinner. For years, Denisovans were known mostly through their genome, not through a large collection of bones. That makes them feel almost like a ghost lineage, a population detected by traces, by comparison, by signals left behind in the bodies of living people. The second layer was stranger. The Tibetan EPAS1 story did not require Denisovans to have lived on the Tibetan plateau in the same way later Tibetans did. The source could have been Denisovan, or Denisovan-related population that carried similar DNA. Ancient DNA from Asia remains sparse, and scientists still refine the map as new remains are sequenced. That caution matters. The evidence points strongly toward Denisovan-related introgression. It does not mean every detail of the ancient meeting is known. It does not tell us the language those people spoke, what they believed, what they called themselves, or exactly where the crucial contact happened. The gene gives a record of inheritance. It does not give a diary. Still, the central meaning is hard to avoid. A fragment from an extinct branch appears to have entered modern humans, survived through generations, and later became useful under the brutal oxygen conditions of the plateau. The Tibetan EPAS1 variant was not merely old. It was likely borrowed from a vanished human world, then preserved because it helped living bodies endure. This is where the title becomes precise. The Denisovan gene shouldn't exist only under the clean model, the model where extinct humans disappeared behind a sealed wall and modern humans adapted alone. Biologically, there is nothing impossible about inherited DNA surviving from archaic humans. The surprise is that the fragment did not remain a meaningless relic. It became part of a working survival system. A vanished population helped write a response inside living lungs and blood. That is not mythology. It is not resurrection. It is not the return of Denisovans as a hidden people. It is inheritance doing what inheritance does, carrying old fragments into new conditions, where selection decides what remains. The extinct source explains the DNA. The modern consequence explains why the genome kept it. Natural selection can move quickly when the environment is severe. On the Tibetan plateau, thin air is not an occasional stress. It is the background of life. It affects sleep, work, pregnancy, childhood growth, and the daily demands of walking, carrying, herding, farming, and breathing. A small advantage can matter there. If a version of EPAS1 helped regulate oxygen without pushing hemoglobin dangerously high, people carrying it may have had better odds over generations. The advantage would not need to be dramatic in every individual. It only had to affect survival and reproduction often enough for the gene to become more common. Pregnancy is especially important in high-altitude adaptation. A developing fetus depends on oxygen delivered through the mother's body. At elevation, that delivery becomes more difficult. Researchers studying highland populations have long been interested in birth weight, maternal oxygen transport, and the risks that thin air creates before a child ever takes a first breath. EPAS1 sits near that larger story. It is not a magic switch that solves every problem, and it should not be used as medical advice. A person's health at altitude depends on many genes: environment, age, history, nutrition, and medical conditions. But as part of a population-level pattern, EPAS1 shows how a genetic variant can become important when survival pressure is constant. The hospital form becomes more than a form. A hemoglobin reading can look ordinary: a number, a unit, a comparison range. An oxygen saturation meter can clip onto a finger and show a percentage. An Ancestry report can reduce deep history into colored regions on a screen. Each object looks modern, clean, and practical. Behind those objects is older evidence. Consumer DNA tests usually do not explain these deep functional stories well. They may estimate ancestry categories, compare relatives, or point to broad regions. They often do not tell a person that one stretch of DNA may have survived because an ancient population met another ancient population, and thousands of years later, the fragment helped bodies handle a particular environment. What your DNA test missed may not be a country, it may be a function. The meaning also has limits. Having the adaptive EPAS1 variant does not make someone more Tibetan in a moral or cultural sense. It does not grant identity by itself. It does not replace language, family, community, history, or lived belonging. Not having it does not erase ancestry, and carrying it does not turn a person into a symbol. Genes are evidence. People are not reducible to evidence. That distinction keeps the story from becoming a purity myth. The Tibetan EPAS1 haplotype matters because it shows mixture, not separation. It shows that a population can carry a useful inheritance from contact with another branch of humanity. It shows that adaptation can depend on ancient entanglement, not isolated perfection. Imagine a mountain settlement at dawn. Smoke rises from a roof. Animals move along a track cut into the slope. A child runs at an elevation where a visitor might struggle to climb stairs. In a clinic, blood is drawn into a tube. In a lab far away, that tube becomes data on a screen. The ordinary scene carries an ancient signature. The blood reading is not only about the present moment, it can carry a message from an extinct human world, filtered through generations of survival on one of Earth's highest inhabited regions. That message is easy to misunderstand if it is forced into the old tree. Draw the human family tree as clean, separate branches, and the Denisovan gene in Tibetans looks like a contradiction. One branch vanished, another branch survived. The vanished branch should not still be shaping a living body's oxygen response. But the genome was never that clean. The branches crossed. Modern humans met archaic humans. Children were born. Some inherited fragments faded away. Some remained as quiet markers. A few, under the right pressure, became useful enough for natural selection to amplify them. EPAS1 is one of the clearest examples because the effect is tied to a visible human challenge. Thin air is not abstract. It can be felt in the chest. It can be measured in blood. It can shape who thrives at altitude and who struggles. The gene links an ancient contact event to a modern physiological pattern. That is why the story still has force. The Denisovan-related fragment did not survive because it was pure. It survived because mixture gave natural selection something to work with. It did not make Tibetans separate from humanity. It tied them to a deeper human record, one where extinct groups were not simply erased from the biological story. The genome remembers contact. In the case of EPAS1, that memory is written through oxygen. It sits in the response to thin air, in the regulation of hemoglobin, in the difference between one highland adaptation and another. It turns the body into an archive entry, not of legend, but of inheritance. The opening contradiction now looks different. A Denisovan survival gene still lives in Tibetans, because Denisovans did not vanish in the simple way the old model suggested. Their bodies disappeared, their communities disappeared. Their names, if they had names in the way we understand them, are gone. But some of their DNA moved forward. In Tibetans, one inherited fragment became part of a survival system for the plateau. In other populations, other fragments tell other stories, some useful, some neutral, some still poorly understood. Ancient DNA keeps showing the same uncomfortable lesson. Human origins were not a clean march of replacement. They were contact, mixing, loss, and adaptation. That does not make the story smaller. It makes it more human. There is no need to turn Denisovans into a legend walking behind every mountain ridge. There is no need to turn EPAS1 into a badge of superiority or a test of identity. The evidence is already powerful without that distortion. An extinct population left a trace. A harsh environment made the trace matter. Living bodies carried it onward. That is the part most simplified ancestry stories miss. They ask where people came from, as if the answer must be a single place, a single label, a single pure line. The EPAS1 story gives a different answer. Sometimes ancestry is a meeting. Sometimes survival depends on a fragment inherited from people who no longer exist. The title says the gene should not exist. The genome answers more carefully. It should not exist if the human family tree is drawn with sealed branches and clean endings. It does exist because the real archive is messier, older, and more intertwined than the textbook diagram allowed. No vanished people returning as myth, no superior bloodline, no purity story hiding inside science, only a trace of ancient contact carried through generations, helping living bodies endure one of the hardest environments on Earth. That is why careful genetic history matters. Ancient DNA can disturb simple stories, but it can also protect us from worse ones. It can show mixture where people imagined separation. It can show adaptation without hierarchy. It can show survival without turning biology into destiny. Subscribe to the Genome Archive for evidence-led stories about ancient DNA, lost lineages, inherited traits, and the parts of the human record most people never learned to read. The Denisovans vanished from the world, but not completely from the human genome.